To appreciate the evolutionary trajectory that shaped mammals into their diverse forms today, we must consider some unconventional, yet plausible scenarios that may have driven key adaptations in our distant ancestors. Over 200 million years ago, in a Mesozoic world dominated by reptiles, early mammaliforms were emerging. Facing a dynamic environment of climatic extremes and high predation pressures, these organisms underwent significant evolutionary transformations, driven by selective forces that demanded both physiological and genetic innovation. One intriguing hypothesis for the evolution of core mammalian traits, such as hair, milk, and live birth, centers on the idea of internal egg retention under harsh conditions. Imagine a primitive mammal-like reptile during an ice age or other extreme cold periods, where freezing temperatures sealed the egg chute shut, leading to the retention of the egg within the body. This scenario, while speculative, may have created an evolutionary bottleneck, forcing adaptations to mitigate the stresses associated with internal egg retention, such as inflammation, immune responses, and metabolic reallocation. Animation of internal egg retention and adaptive response. In response to an egg stuck inside the body, the most immediate adaptive mechanism would involve inflammation and localized immune activity. The accumulation of immune cells and fluid around the egg could provide a rudimentary cushion to relieve mechanical stress on the tissues and protect against infections. Over evolutionary time, such inflammation-associated responses could have driven the development of specialized glandular structures to secrete immune-modulating and anti-inflammatory substances, initiating the rudimentary stages of gland evolution in these early mammals. Diagram illustrating gene adaptations in gland evolution. At the genetic level, this transition would likely involve the repurposing of genes associated with the immune system and wound healing. Antimicrobial peptides, such as lysozyme and lactoferrin, which are already conserved across vertebrates, could become increasingly expressed in these nascent glandular structures. Their roles would extend from merely controlling infections to forming the molecular basis for nutrient-rich secretions. This repurposing would not have been an instantaneous process, but rather an accumulation of mutations, gene duplications, and regulatory changes favoring the expression of these molecules in newly forming glandular tissues. Scene transition, early mammal adapting hair structure. Simultaneously, the retention of an egg and the associated metabolic costs would shift energetic priorities, potentially reducing the selective advantage of maintaining large, energetically expensive feathers. In extremely cold environments, the finer tips of feathers would be prone to structural damage due to freezing, making them less effective for thermoregulation. This could lead to a transition toward hair-like structures, which would allow for more efficient heat retention through denser coverage and a greater number of insulating filaments per unit area. Here, the underlying genetic adaptations would involve shifts from beta-keratins, which form the rigid structure of feathers, toward alpha-keratins, which are better suited for the pliability required in hair formation. Genomic studies indicate that hair and feathers share deep homology in terms of keratin gene families, yet they diverge significantly in terms of their functional deployment in integumentary structures. In the context of hair evolution, the activation of hair follicle morphogenesis genes, such as EDAR and FOXY3, may have played a pivotal role in transitioning from sparse, reptilian scales or feathers to densely packed hair. These genes regulate follicular density and hair patterning, providing a genetic foundation for adaptive responses to cold stress. Gland evolution and milk development. As glandular structures became more sophisticated, a convergence of evolutionary pressures likely facilitated the transition from infection control to nourishment. Genetic evidence supports that casein gene families, which underpin milk's nutritional properties, arose from calcium-binding gene duplications approximately 200 million years ago. This supports the notion that initial glandular secretions may have been modified to enhance calcium transfer for egg retention, which later evolved to fulfill the nutritional demands of live-born offspring. The milk of modern mammals, enriched with components like immunoglobulins and lactoferrin, reflects a genetic legacy of immune modulatory proteins being co-opted into lactation, thus providing both nourishment and passive immunity. Scene: phylogenetic diagram depicting evolutionary transitions. The implications of this hypothesis extend beyond glandular and integumentary evolution. The emergence of complex milk composition, as driven by the interplay between nutritional and immune demands, highlights an intricate gene regulatory network that has been finely tuned over evolutionary time. Comparative genomics reveals that different lineages of mammals, such as marsupials and eutherians, exhibit distinct but convergent adaptations in milk-related genes, reflecting lineage-specific selection pressures. This underscores the plasticity of gene regulatory mechanisms in adapting to diverse ecological niches and reproductive strategies. Diverse modern mammals with specialized hair and milk. The diversification of mammals following these evolutionary milestones is evident in the remarkable variation in hair density, structure, and function across species, as well as in the diversity of milk composition tailored to different environmental challenges and offspring needs. For example, mammals in polar environments evolved milk with higher fat content to support thermogenesis in young, while those in arid climates developed milk with lower water content to reduce dehydration risks. Closing scene integrating evolutionary and genetic perspectives. Thus, the evolutionary history of mammals is one marked by profound genetic and physiological innovation. The speculative scenario of internal egg retention due to environmental stressors like freezing offers a unique lens through which to view the origin of mammalian traits such as hair and milk. By integrating genetic evidence with hypotheses about environmental adaptation, we gain a deeper understanding of how natural selection shaped these traits to give rise to one of the most adaptable and diverse lineages in the animal kingdom. The hypothesis, while unconventional, challenges us to think critically about the interconnectedness of seemingly disparate adaptations, how metabolic costs associated with reproductive strategies, integumentary evolution, and glandular development may have intertwined to produce the unique biology that defines mammals today. By analyzing the genetic legacy encoded in our genomes, we uncover a story of resilience and adaptive versatility that underlies the success of mammals in occupying almost every terrestrial and aquatic habitat on the planet.